Assalamu alaikum warahmatullahi wabarakatuh. Good morning or good evening, everyone. I hope that you're doing fine and everything is okay. Today we will start the urinary system, the histological structure of the urinary system. In today's lecture, we will study about the urinary system, including first of all the kidney. Okay, we will start by the kidney itself, and then we will go to the uriniferous tubules. Okay, the tubules that contain urine in it, and then we will go to the Bowman's capsule, and then we will study a cell that is called podocyte, and lastly we will go through the glomerular capillaries present that delivers the blood to the Bowman's capsule, to the structure, the corpuscle. Okay, now the kidney appear as a bean-shaped structure. Everybody have two kidneys, of course. It's formed of outer part that is called the medulla. All of the outer part. This is the kidney itself. The outer part of it is called the cortex, and then the inner part here. This part is called the medulla. Okay, then the outer part is called the cortex, and the inner part is called the medulla. Okay, the medulla appear light gray, and it is formed of renal pyramids, and their apex is directed towards the renal pelvis. And these are the renal pyramids, and the apex is directed towards something called the renal. It is directed towards something called the renal pelvis. Okay, this is the renal pelvis. All of this structure, all of this area is called the renal pelvis. So the medulla formed of pyramidal structures, pyramids towards the renal pelvis. Okay. Again, as you can see here, this is the renal, the medulla of the kidney, and these are the, this is the medulla, and the outer part is the cortex. The medulla is formed of renal pyramids. The renal pyramids open to a minor calyx, and the minor calyx join together to form the major calyx. Okay, and the cortex and the medulla surround, and the medulla underneath this area of the cortex, and the medulla underneath this part is called the renal lobule. Okay, and you have renal columns, the part that resembles the cortex, that is present in between two medullary areas, medullary rays, medullary columns. Okay, so, and the renal lobule is formed of medullary ray and is surrounded by cortical tissue. This is the medullary ray, and this is the cortical tissue. Anyway, the outer part is called the cortex, and the inner part is called the medulla. Of course, histologically speaking, the kidney is formed of stroma and parenchyma. Stroma formed of a capsule. You have a capsule surrounding the kidney, and you have reticular connective tissue supporting the parenchyma. The parenchyma are formed of the uriniferous tubules. Again, in this slide, this is the renal pelvis. You have the outer part is the cortex. You have medullary pyramid open into minor calyx. Minor calyx join together to form the major calyx. Okay, and you have some cortical tissue extend in between the medullary rays or the medullary tissue or the medullary pyramids. You have some cortical tissue extend in between them. Okay, and it is called the renal column. This is the renal loop formed of some cortex, part of the cortex and part of the medulla. And this is a medullary ray. Okay. For the uriniferous tubule, the uriniferous tubule is formed of renal corpuscle, okay, and it is formed of three things. And then you have the longest structure, the proximal convoluted tubule, and then you have the loop of Henle, and then the distal convoluted tubule, and lastly the collecting tubule. Okay. So the nephron is divided into renal corpuscles and renal tubules. Here again, this is the renal the glomerular corpuscle. It contains the glomerulus. The glomerulus is formed of blood vessels. The blood vessel here, as you can see, enter in this area, and then divide, forming lots of capillaries, and then they exit from this part. Most of the plasma, most of the fluid inside the blood is filtrated towards the outside here. Most of the fluid in the blood filtrated into the tubules, and it passes all through the proximal and distal tubule, proximal loop of Henle and then distal tubule. And all through this area, you can see you have renal reabsorption, and you have also some secretion. Okay, and then urine is formed and concentrated in the collecting tubules. Here again, you have blood vessel, artery and vein. Okay, artery and vein. The artery forms the glomerulus here in this area, okay, and the blood is filtered through the proximal convoluted tubule, then go to the loop of Henle, go again to the distal convoluted tubule, and then to the collecting tubule. Okay, there is reabsorption all through these areas, and urine is finally formed and secreted into the renal pelvis through the renal papillae. For the development of the renal corpuscle or the Malpighian corpuscle, at first there is a blood capillary, and some epithelial cells. The blood capillary push on the epithelial cell, pushing them inwards, forming something like a balloon. Okay, the balloon has two surfaces, the outer surface, this area, and you have another inner surface, this part. Okay, the outer area is called the visceral port and the parietal port, and the inner is the parietal port. And then the blood vessels that invaginate into the epithelial layer would create lots of loops and capillaries. Okay, creating to increase surface area, to increase the surface area for filtration, for blood filtration. Okay, and all of the filtered parts, all of the things that are filtered would go into this space, and then they would pass into the proximal convoluted tubule to be concentrated, reabsorbed, and some tubular secretion would happen. Here again, in this slide, as you can see, from the beginning you have a vascular area and you have a urinary area. Vascular area, blood vessels, and another area for urine. This area is called vascular part, vascular pool. Okay, vascular pool, whereas the other pool is called the urinary pool. Okay, vascular pool and a urinary pool. Here again, as you can see here, you have an afferent. The entering blood vessel is called an afferent blood arteriole, and you have an efferent for the exit, and in between them you have lots of capillaries, and any capillary is lined by endothelial cell, of course. Okay, so you have afferent arteriole, and you have efferent arteriole, and you have blood capillaries in between. So, and you have, this is the vascular pool, and this is the urinary pool. Okay, and the renal corpuscle or the Malpighian corpuscle is formed of three structures. The first structure is the Bowman's capsule or the balloon itself. This is this structure, this blue or green structure. This is the first part, and this is called the Bowman's corpuscle. Okay, and then you have the blood capillaries, Bowman's. Okay, this is the first part, and then you have the blood capillaries, and the third structure is called the mesangium. The mesangium we will discuss in the next lecture. The mesangium. Okay, so the renal corpuscle is formed of three structures. They are the Bowman's corpuscle and the glomerulus and the mesangium. Okay, for the Bowman's capsule or the Bowman's corpuscle, it is a double-walled structure. Double-walled structure, as we have said, it is like a balloon, an invaginated balloon. Okay, and you have lots of blood capillaries in this area. Okay, invaginated blood capillary. This part is the urinary pool, whereas this part is the vascular pool. In this H&E slide, you can see the glomerular capillary lumen. All these empty structures, these are the glomerular capillaries. Okay, this is the vascular pool, and this is the urinary pool. Okay, and you can see lots of yellowish structures. These are the endothelial cell or podocyte or mesangial cell, either of these three cells. These are them, of course. Okay, so this is the Bowman's capsule itself. The Bowman's capsule is this area. Okay, and the cells lining the podocytes that are lining the blood capillaries in this area. The outer port is called, of course, the parietal port, and the inner port is called the visceral part. The outer pool, this area is called the parietal. The parietal layer. And the other one is called the visceral part. layer. Okay, so you can, as you can see here, you have a parietal layer and you have a visceral layer. The visceral layer is lined by a cell that is called podocytes. Okay, so you have the parietal and you have the visceral, and the visceral layer is lined by a cell called podocyte, whereas the parietal layer is lined by just simple squamous epithelium, and it just separates the Bowman's capsule from the surrounding. That's all. Here again, as you can see in this slide, the blood capillaries that enter the blood through is called the afferent arteriole, and the blood vessel from which the blood exits it is called the efferent arteriole. Okay, and you have lots of capillaries in between. You have, this is the parietal pool, the parietal layer, and the other one, the podocyte, is called the visceral layer of the Bowman's capsule. Okay, and this dark space, this is where the filtered blood enters, and it goes to the urinary pool of the proximal convoluted tubule. Okay, for the podocytes, they are modified epithelial cells. They should be simple squamous epithelium like the parietal part of the Bowman's capsule, but they change and form something else. They form a structure that have pedicles or have processes. These are the pedicles. Okay, this formed of a cell. This is the cell, and the cell send lots of arms, and each of these arms form a secondary structures, okay, secondary processes that line the capillaries. Okay, so this is the blood capillaries. The blood passes in this area, pass through here, and the podocytes line all of the blood capillaries, okay, to form the blood-renal barrier, something called the blood-renal barrier. Here again, the podocyte should be a squamous epithelium. As you can see here, they are long and flat, okay, but they send lots of processes, primary process and then secondary process, to cover all of the blood capillaries present in the glomerulus. They are separated from the glomerulus, from the blood capillary, by a space called subpodocytic space. We will see it just in a minute. Okay, here I guess you can see here you have three layers: the podocyte, and then the endothelial cell. But this podocytic process, they are the secondary podocytic structure, secondary process. Okay, and this is the endothelial cell, and in between here you have the basement membrane. Okay. So again, you have, this is the podocyte. You have a primary process. From the primary process, it sends secondary process, and the space the space that is present in between here, this space that I'm circling, this is called the subpodocytic space, the space between the podocyte and the blood capillary itself. Okay, and here you have a fenestrated blood endothelial cell, a fenestrated simple squamous epithelium, and you have a basement membrane, fenestrated endothelial cell, then basement membrane or basal lamina, and pedicles, which are the secondary process of the podocytes. Okay, again, the secondary process of the podocytes is separated from each other by a space, another space. Okay, this space is called a filtration slit. Filtration slit. Okay, a hole, filtration hole, and this slit is covered by filtration slit diaphragm. It's covered by a diaphragm. You can see here in the endothelial cell you have a fenestrated endothelial cell. It is called fenestrated. It has filtration slit, but it is not covered by any diaphragm. Whereas the secondary process or the pedicles of the podocyte have a filtration slit and it has the diaphragm. Okay, the filtration slit is about 25 nanometer, whereas the thickness of the diaphragm is about 5 nanometer in diameter. Of course, for the podocyte to appear as this structure with primary and secondary process, it must contain lots of microfilament and microtubule in order to form the cytoskeleton of the cell. The cell itself must have a very strong cytoskeleton. Here again, this is the podocyte itself. Podocyte and another podocyte. Okay. And this area is enlarged here. You have, this is the secondary process. You have a filtration slit, and you have a filtration slit diaphragm that is about 5 nanometer in diameter. Again, a large podocytic process, secondary process, and you have a filtration slit, and you have a filtration slit diaphragm in between. Whereas from the capillary side, from the blood vessel, you can see this is the simple squamous epithelium or the endothelial cell, and you have no slit diaphragm in between, no diaphragm. And the space in between the podocyte and the endothelial cell, the secondary process of the podocyte and the endothelial cell, is called the Bowman's membrane. Okay, and the blood is filtered from here to here, and then it passes the filtrate would pass till this space in the Bowman's corpuscle, and then it would go to the urinary pool, to the proximal convoluted tubule. Here again, this is a scanning electron microscope. This is the podocyte, primary process, secondary process. As you can see, there is a little space, a small space in between the secondary process each other. Okay, this is called the filtration slit, and it is covered by a diaphragm. Okay, here again, as you can see, this is a podocyte and another podocyte. You have primary process, secondary process. This is the blood capillary. Okay, this area is called the blood renal barrier, and it is formed of three structures: the endothelial cell, the secondary process of the podocyte, and the basal lamina or the basement membrane of the that is formed from both of them together. The podocyte by electron microscope, of course, it contains some rough and Golgi and ribosomes, and it have a very good cytoskeleton like microtubule and microfilament. And as you can see, the nucleus appear irregular. Okay, irregular nucleus and very strong cytoskeleton. What about the function of the podocyte? The podocyte share in the formation of the glomerular basement membrane and the blood-renal barrier. Okay, for the glomerular capillaries enter from the afferent arteriole, as we have said, afferent arteriole, and then they form the capillaries, the glomerular capillaries, and go out through the efferent arteriole. Okay, you have the Bowman's capsule. This is the space from which the fluid would go to the proximal convoluted tubule. Okay, so the afferent contain non-filtered blood, and it would branch to about 50 glomerular capillaries, and it would go to the efferent. The efferent contain filtered blood. Okay, the endothelial cells are fenestrated, as we have seen in the previous slide, and these fenestrae are not covered by diaphragm, whereas the fenestrae between the secondary pedicle of the podocyte are lined by diaphragm, which was 5 nanometers in diameter. Okay, here again, this is the blood capillary, and these are the endothelial cells. Okay, the filtration slit or the fenestrae in the blood endothelial cell is not covered by a diaphragm, whereas the fenestrae in between the secondary process of podocytes are covered by diaphragm. The filtration slits are about 25 nanometers in diameter, whereas the diaphragm itself is about 5 nanometers in diameter. This area is the basement membrane or basal lamina. The basal lamina is formed from both the endothelial cell and the secondary process of podocyte. Okay, as you can see here, you have an electron-lucent port, and in between you have an electron-dense area. Okay, these are the three structures of the basement membrane or the basal lamina. Here you have 2 electron-lucent port and 1 electron-dense port. This is a very good slide. Okay, this is the endothelial cell, and you have all of these are the remaining of the endothelial cell. It forms like a sieve. It forms something like a sieve. It have many fenestrations, and these fenestrations are not covered by a diaphragm. Okay, and then you have on the other side, this is the podocyte, and this is another podocyte. Okay, and you have here another one. Okay, all of these are the visceral part of the Bowman's membrane. Okay, all of these are podocytic structures, podocytes. Okay, the podocyte has primary process and have secondary process. The secondary process are separated from each other by filtration slits that are covered by a diaphragm. Okay, and in between you have the basal lamina. This is the basal lamina. Okay, the basal lamina is formed from the endothelial cell and from the podocyte. okay, from the endothelial cell and from the podocyte, okay, and the three of them, the endothelial cell, the basal lamina, and the podocyte, form the blood-renal barrier, and this barrier is affected in many pathological diseases of the kidney. Okay, if any compensation, any damage happen in the blood-renal barrier, there would be increased in the filtration, and the plasma would plasma proteins would go in, would be released in the urine, and causing different diseases, different pathological diseases. Okay. So what are the two parts that form the uriniferous tubule? Okay, what are the two parts that form the uriniferous tubule? The uriniferous tubule was formed of something like that: primary, hypophthalmic, secondary, and then primary, hypophthalmic, and second distal convoluted tubule, and then collecting tubule. Okay, this is the Bowman's corpuscle, and you have here afferent and efferent arteriole. Here's lots of blood capillaries in this area. So the afferent arteriole, what was the afferent arteriole contain? Afferent arteriole contained unfiltered blood, whereas the efferent contained filtered blood, and this area is called the vascular pool, and this area is called the urinary pool. Okay, and then you have here, this is the proximal convoluted tubule, lupus, distal convoluted tubule, collecting tubule. All of these are the structure of the uriniferous tubules. Okay, and this structure itself was called the Malpighian corpuscle, or the urinary renal corpuscle, Malpighian corpuscle, or the renal corpuscle. The renal corpuscle formed of the Bowman's capsule, the glomerular capillaries, and the mesangium. The mesangium will be discussed in the next lecture. Okay, so you have Bowman's capsule, glomerular capsule, glomerular capillaries, and the mesangium. These are the renal corpuscle, whereas the uriniferous tubule is formed of the nephrons and the collecting tubules. Okay, what about the Malpighian corpuscle? Oh, I have already said it. What is the Malpighian corpuscle formed of? The renal corpuscle. I'll give you a minute. I'll give you, just pause the video and think about it. What was the structure of the Malpighian corpuscle? Okay. The Malpighian corpuscle was formed of the, what is this structure that I'm drawing? Yes, you're right. This is the Bowman's corpuscle. Okay, it is formed of parietal port and visceral port. This port is formed of simple squamous epithelium, whereas this port is formed of what? Yes, it is formed of podocytes. And then you have arteries. Okay, formed of many capillaries, divided into many capillaries in here in this area. The entering artery is called afferent arteriole, and the going out or the exit is called the efferent arteriole. Okay, so it is formed of number 1, the Bowman's corpuscle. Number 2, the glomerular capillary. And number 3, the mesangium. Okay. This forms the Bowman's corpuscle, glomerulus, and the mesangium. Okay. I hope that you have find this lecture. Again, in this slide, this is the renal pelvis. You have the outer port is the cortex. You have medullary pyramid open into minor calyx. Minor calyx joined together to form the major calyx. Okay, and you have some cortical tissue extend in between the medullary rays or the medullary tissue or the medullary pyramids. You have some cortical tissue extend in between them. Okay, and it is called the renal column. This is the renal loop formed of some cortex, part of the cortex and part of the medulla. And this is a medullary ray. Okay. For the uriniferous tubule is formed of renal corpuscle. Okay, and it is formed of three things. And then you have the longest structure, the proximal convoluted tubule. And then you have the loop of Henle, and then the distal convoluted tubule, and lastly the collecting tubule. Okay, so the nephron is divided into renal corpuscles and renal tubules. Here again, as you can see in this slide, the blood capillaries that enter the blood entered through is called the afferent arteriole, and the blood vessel from which the blood exits it is called the efferent arteriole. Okay, and you have lots of capillaries in between. You have, this is the parietal pool, the parietal layer, and the other one, the podocyte, is called the visceral layer of the Bowman's capsule. Okay, and this dark space, this is where the filtered blood enters, and it goes to the urinary pool of the proximal convoluted tubule. Informative for you, and you have understood each and every part of it. If you didn't understand anything, please please read the lecture from your textbook, from your books, and ask us anything anytime in the department, in the histology department. And thank you very much.